Charles Mazenga, and I'm here to present a topic called lipid oxidation and cancer. So I'd like to explore the relationship. So lipid oxidation is a process where lipids undergo chemical degradation, and it has been implicated in various diseases including cancer. So this presentation delves into the intricate relationship between lipid oxidation and cancer, exploring the mechanisms, potential consequences, potential strategies for intervention. By understanding how lipid oxidation contributes to cancer development, we can gain valuable insights for prevention and management. Begin, I would like to acknowledge that there are several different theories that scientists and people talk about to explain the relationship between lipid oxidation and cancer. So just to give you an example of one, there's a review study by Jang A. Kim in 2020 that elaborated stated that some cancer cells rely on lipid peroxisomal metabolism for energy. So this is just an example of one theory that people talk about to explain this relationship. So in this presentation, we'll be discussing and exploring other theories to explain this relationship. So to begin, lipid oxidation plays a crucial role in energy production within the human body. When fats, triglycerides, are broken down through a process called lipolysis, they are converted into fatty acids and glycerol. These fatty acids undergo lipid oxidation, also known as beta oxidation, in the mitochondria and in the peroxisomes to generate ATP, the energy currency of cells. Now, lipid oxidation in the peroxisomes generates peroxides, a reactive oxygen species, H2O2. It's like a byproduct metabolite. However, peroxisomes use catalase enzyme to convert H2O2 to water and oxygen to prevent oxidative stress. Now, I would love to emphasize on this highlighted area on the slide that says lipid oxidation in the peroxisome generates peroxides, reactive oxygen species area H2O2. Now, why is this important? So I want to emphasize this because one of the theories that people talk about to explain the relationship between lipid oxidation and cancer is through lipid peroxidation. Now, remember I said that the peroxisomes have the ability to convert the reactive oxygen species to water to prevent oxidative stress. Now, when the peroxisomes loses the ability to convert H2O2, the peroxides, into water, then lipid peroxidation occurs. Now, what is lipid peroxidation? Lipid peroxidation is an oxidative degradation process of lipids. It starts with the reactive oxygen species that attack long-chain polyunsaturated fatty acids by getting a hydrogen atom from them. So the fatty acids that are more prone to lipid peroxidation are long-chain fatty acids from 18 to 22 or more carbon chains items. So just to give you an understanding of lipid peroxidation, the process. So as I mentioned, it involves a reactive oxygen species. So it has three processes. Initiation, the process begins when a free radical such as a hydroxy radical, the ROS that we talked about, abstract a hydrogen atom from a lipid molecule, generating a lipid radical. The second step is propagation. The lipid radical reacts with a molecule oxygen to form a peroxide radical, which then abstracts a hydrogen atom from another lipid molecule, perpetuating the chain reaction, generating more lipid radicals and escalating the oxidative process. Now, this propagation period is the stage that induces oxidative stress. So oxidative stress is mostly occurs because of this propagation stage. The third stage is now termination. The chain reaction can be terminated by reaction of two radicals forming a non-radical product or by the reaction of a radical with an antioxidant, which neutralizes the radical by donating electrons to stabilize free radicals and interrupts the chain reaction. Now, like to emphasize the propagation period, which induces the oxidative stress, and also like to bring to your attention for you to keep in mind the antioxidant that counter this reaction. So it terminates the lipid peroxidation and most often cancels the oxidative stress. To explore further the theory that lipid oxidation affects cancer through lipid peroxidation and oxidative stress. Stress is an imbalance between the production of reactive oxygen species and the ability of the body to detoxify them. Big role in cancer development. So if you remember, we talked about how the peroxisomes have the ability to detoxify, to convert the reactive oxygen species. So now once the peroxisome loses this ability, then oxidative stress can occur through lipid peroxidation. So a study in 2015 by Marco and other authors, oxidative stress denotes high levels of ROS, and ROS can damage DNA, proteins, lipids, leading to cellular dysfunction and uncontrolled cell growth. Now, other mechanisms that support this theory are through DNA damage. So reactive oxygen species can directly damage DNA and induce mutations, leading to activation of oncogenes or inactivation of tumor suppressor genes, which can drive cancer development. And this was stated and proven in studies by Iqbal MJ in 2024 and other studies in 1994 by Zhu MQ and other authors. A second mechanism to support this theory is through cellular signaling. So reactive oxygen species can disrupt cellular signaling pathways involving cell growth, proliferation, and apoptosis, promoting uncontrolled cell growth and tumorigenesis. Now, this was elaborated in a study by John M. Kim in 2020, and another study by Singh F. and other authors in 2022, another study by Seth T. and other authors in 2006. And another mechanism to support this theory is through inflammation. So reactive oxygen species can trigger chronic inflammation, a key contributor to cancer development. Inflammation can create a favorable environment for tumor growth by promoting angiogenesis, cell proliferation, and metastasis. And this is also elaborated and stated and proven in a study in 2022. Patient can fight cancer through lipid peroxidation by the reactive oxygen species to induce oxidative stress. Now I've talked about the reactive oxygen species and how it promotes or induces cancer development. Now, so I'd also like to bring to you another theory that lipid peroxidation can also be related to cancer prognosis. So there is a study of clinical implications of lipid peroxide levels in plasma and tumor tissues in breast cancer patients by Talita B. and other authors in 2022. So it investigated the lipid peroxidation profiles in tumor and plasma samples from breast cancer patients. So the number of the sample size was 140, and they discovered that high LPA levels were found to be in correlation with or to be related to poor prognosis. So here on this page, on this slide, you can see different p-values of premenopausal patients, the p-value of 0.012 in tissues, high-grade tumors, metastatic disease, chemo-resistant tumors. So they reviewed a number of factors which all had a p-value lower than 0.05, which indicated that there's a significant relationship between lipid peroxidation profiles and poor prognosis. So the higher the profiles, then the lower the prognosis. And this was in high-grade tumors, chemosynthetic tumors, metastatic disease, but in breast cancer patients. So this is just another study. This is just a study to give an insight of how lipid peroxidation can affect cancer prognosis. Now, moving on. So apart from the reactive oxygen species, the peroxides produced through beta oxidation, the peroxisome, there are also other metabolites products like MDA and HNE that are produced when lipid oxidation occurs. So I would also like to talk about how these products also affect cancer initiation, progression, and metastasis. So lipid oxidation has been implemented in both initiation, progression of cancer. Research suggests that lipid oxidation products can promote cancer development through various mechanisms. So a study by T.W. Kensler and other authors in 2019 elaborated how these products can affect the different cancer stages. So firstly, it can affect at the initiation stage. Lipid oxidation products such as melanoialdehyde, MTA, and 4-hydroxyl-nonenal HNE can damage DNA, leading to mutations that contribute to cancer initiation. The second stage, progression, lipid oxidation products can promote tumor growth and metastasis by enhancing angiogenesis and promoting cell proliferation and inhibiting apoptosis. So, just to talk about a little bit about the progression. So, to receive blood, the cancer cells, like any other cells in the body, can send out chemical signals that cause angiogenesis to begin. In this case, angiogenesis is harmful to the human body because it will promote the cancer growth. So it's creating blood vessels that feed the tumor with oxygen and nutrients. The tumor then begins to grow and cancer cells can spread elsewhere in the body. Now, angiogenesis is not always a bad thing because other normal cells also need this. But in this particular situation, it is harmful because it is feeding the tumor cells. Now, the third stage, metastasis. Lipid oxidation products can contribute metastasis by inducing EMT, a process that allows cancer cells to spread to surrounding tissues and spread to distant sites. Okay, so exploring further about how these other products, MDA and HNA, affect cancer. Now, there's another theory that these products can have an effect on cancer risk. So studies have shown that increased lipid oxidation is associated with elevated risk of developing various cancers, including colorectal, breast, and prostate cancer. So a study by Gaussman and other authors in 2012 highlighted the different lipid oxidation products and their mechanisms and how they affect specific cancer types. So, for example, as you can see in this slide, colorectal cancer is the risk is influenced by MTA and HNE through the mechanism of DNA damage and inflammation. And as we talked about in the previous slides, how inflammation can cause cancer progression, cancer development. And then in breast cancer as well, the associated products of lipid oxidation are MTA and HNE, and they can increase the cancer risk by promoting cell proliferation and angiogenesis of tumor cells. And the same products also apply to prostate cancer, and the risk is increased because MDA and HNE promote cell growth and metastasis. We have talked about how lipid oxidation, peroxidation, ROS, and other products like MNA and HNE, how they affect cancer development, progression, metastasis, the cancer risk and as well as cancer prognosis. Now I'd like us to talk about how lipid peroxidation can also affect immune cells during cancer. So this is I'd like to talk about a study by Li-Ling Zio. It was done in 2024. So it gives us a tandem cycle of how lipid peroxidation can be good and bad at the same time. So let's start with immune cell lipid peroxidation. So in the immune cells, when lipid peroxidation occurs, it inhibits the body's capability to fight off the cancer growth. So for example, it impairs the anti-tumor effect of CD8 T cells and NK T cells. So it decreases antibody production of B cells, inhibits antigen cross-presentation of DC, and inhibits survival of M2 macrophages, and a number of other mechanisms of how lipid peroxidation in immune cells can influence or promote cancer cells by inhibiting the capability of our immune system. Now, so the inducers of lipid peroxidation in immune cells can be, for example, the RISL3, Eristin, and some other FDA-approved drugs such as sorafenib and olaparib can also have this effect of lipid peroxidation in immune cells. And inhibitors of this effect can be FER1 and LIP1. Okay, so now let's look at the lipid oxidation in tumor cells. So lipid oxidation in tumor cells can have a positive effect in humans because it inhibits proliferation of tumor cells, it inhibits metastasis of tumor cells, and it induces ferroptotic cell death of tumor cells. And so this slide shows us how lipid peroxidation affects positive and negative effects in our body, and during cancer it can affect in either ways. About lipids, oxidation, peroxidation, all that stuff. So how can we counter this effect of lipid peroxidation, lipid oxidation, the byproducts, and cancer? How do we counter these effects? How do we stop this relationship? Okay, so vitamins and antioxidants counter lipid oxidation effects. If you remember through the previous slides, we mentioned about how antioxidants can have a factor in termination of lipid peroxidation by donating electrons. Okay, so this is one of the mechanisms of how we can stop the oxidative effects. Now, dietary vitamins and antioxidants play a crucial role in protecting against lipid oxidation. These compounds can effectively counter the damaging effects of free radicals, the reactive oxygen species, thereby preventing lipid damage and its associated health risk. Now, this was proven in a study by Cook Anderson and other authors in 2013. Okay, so different vitamins include vitamin C, vitamin E, beta-carotene, and selenium. To start with vitamin C, a potent antioxidant that scavenges free radicals, reducing lipid oxidation and protecting cells from damage. Vitamin E acts as a lipid-soluble antioxidant that protects cell membranes from oxidative damage by neutralizing lipid radicals. Beta-carotene, a precursor to vitamin A, beta-carotene exhibits antioxidant properties, protecting against lipid oxidation and promoting cell health. And selenium is an essential trace mineral that supports the activities of enzymes involved in protecting against oxidative stress and lipid peroxidation. So these are the vitamins that can counter the effect of lipid oxidation. I just like to give an examples of foods that are rich in selenium. So these are nuts, grains, and meat. And foods that are high in vitamin E are sunflower seeds, almonds, hazelnuts, peanuts, salmon, avocados, red sweet peppers, Brazil nuts, a lot of several things. And as for vitamin C is high in citrus fruits. And foods that are high in beta carotene include orange color, includes like most likely have like an orange color, orange hue. Um, so let's look at the foods rich in beta carotene. So sweet potatoes, butternut squash, cantaloupe, mango, canned pumpkin, carrots, and these are the ones that I mentioned that they usually have like an orange hue. And others also include spinach, kale, and collard greens. So we're now focusing on management. So what does this relationship of lipid lipid oxidation have to do with cancer management? Let's discuss about that. So, and the understanding of lipid oxidation's role in cancer opens new avenues for cancer management. Targeting lipid oxidation pathways might offer novel strategies for cancer prevention, treatment, and management. So let's start with prevention. By consuming a diet rich in vitamins and antioxidants, individuals can reduce oxidative stress, protect against lipid oxidation, and potentially lower their risk of developing cancer. I hope you can remember how we talked about cancer, lipid oxidation, peroxidation, and its relation to cancer development, the risk, as well as the progression. So eating foods, a diet rich in vitamins, we talked about vitamin A, E, and selenium can protect against lipid oxidation and lower the risk of developing cancer. I hope we can remember that and appreciate the things we discussed earlier. Now, second, treatment. Antioxidant therapies in combination with conventional cancer treatments may enhance treatment efficacy and minimize side effects by reducing oxidative stress and promoting tumor cell growth. Management. Lipid oxidation biomarkers can be used to monitor cancer progression, assess treatment response, and personalize therapy based on individual patient oxidative stress levels. Okay. So we talked about how regulation of peroxidation can help in the prevention and management, prevention and treatment of cancer. So now I just want to talk about like sort of like contraindication. So when considering the regulation of lipid peroxidation to inhibit tumor progression, it's essential to account for both tumor cells and immune cells. These scenarios can become quite intricate. So if you remember, there was a slide where it showed how lipid peroxidation can affect like either like in a good way or in a bad way to like the immune cells and the tumor cells. So like the effect is different. Lipid peroxidation could be both harmful and beneficial. Therefore, studies like those by Song S.R. and other authors elaborate nanoparticles that selectively deliver lipid peroxidation inducer to tumor cells. So they're only delivered to tumor cells and not normal cells. So this might be a promising way to enhance cancer therapy. As we mentioned, if you promote lipid peroxidation in normal cells, then it promotes cancer growth. But if you deliver lipid peroxidation to tumor cells, then it inhibits the tumor growth. So that's something just to keep in mind to consider. Now, so nanoparticles co-delivering dihydroatomycin and lipid peroxidation inducer RSL3 have been shown to efficiently induce ferroptosis in tumor cells of pancreatic ductal adenocarcinoma, melanoma, and metastatic breast tumors and trigger T-cell-based anti-tumor immunity. So this was elaborated in a study by Liu Ling Xiao in 2024. With all this in mind, what are the practical applications and future directions? The growing body of research on lipid oxidation in cancer holds significant promise for developing effective interventions and improving cancer management. Firstly, personalized nutrition. Developing a personalized dietary recommendations based on individual lipid oxidation profiles could help optimize antioxidant intake and reduce cancer risk. Secondly, through drug development, exploring novel drugs that specifically target lipid oxidation pathways might offer effective treatments for various cancers and potentially with fewer side effects than current therapies. That's good. Thirdly, through prophylactic strategies, so identifying individuals at high risk for lipid oxidation and genetic testing and biomarker analysis could enable early intervention, early detection of cancer development. So I hope that this presentation has increased your awareness of the relationship between lipid oxidation, cancer, and actually the goal of my presentation. I wanted to inform my fellow students on the opportunity we have to study lipid oxidation in cancer and explore the possibilities like how this interaction could improve cancer management. So I hope this has at least pricked your interest into this field. And thank you so much for listening. And these are my references.